My name is Ulrich Broeckel. I'm a faculty member at the Medical College of Wisconsin. I'm a professor in the Department of Pediatrics, also medicine and physiology. And my work really focuses on complex disease genetics, particularly we're focused on cardiovascular diseases. We really have two components in my lab. The one component is I'm a technical director of a clinical diagnostics lab. So we're doing clinical diagnostics to identify genes for complex diseases. And on our research side, our research arm, we're focusing to really understand the functional impact of genome variation in genes on cardiovascular disease, particularly in heart disease, like for example heart failure or pre-stages which lead to heart failure. We use a lot of model organisms, particularly cell culture. We use induced pluripotent stem cells. And so one of our main readouts is really expression. And so RNA sequencing is sample preparation, which goes into RNA sequencing. That's a very big theme for us. The challenge, it's been pretty clear over the last few years, and I think this is going to keep us busy for many years to come, is really to understand the function of this genome variation. How does a mutation, a variant, a structural abnormality contribute to the overall disease mechanism? And I think this understanding the underlying biology, I think, is really going to be absolutely critical, not only for clinical diagnostics, but really also to use then genetic information to develop new treatments, like for example, drug development. If you have model organisms, right, or if you study model organisms, you study cell culture, use cell culture experiments, I mean, the readout gives you very different sources of data. You know, if you look at expression, you look at RNA, you look at microRNAs, analyzing these large data sets is absolutely critical to understand gene function. And so having tools which allow you to take these diverse data sets, take expression, take microRNA, take, for example, proteins, to really combine this and bring this and connect this to potential disease pathways. I mean, that is really the analysis which is necessary to gain these insights. And so I think bioinformatics, anybody who works in genetics and genomics and works in this field, recognizes the tremendous need for bioinformatics and the functional annotation and the analysis of diverse data sets, and platforms which really bring this together are absolutely critical. IPA, I think, is a pretty exciting tool for us to really get a first glimpse of like gene function. So the annotation there is very comprehensive. We can analyze gene expression, we can look at pathways, how certain genes really fall into these pathways, how expression changes as we perturb certain genes, how that affects disease mechanisms. And so that annotation really gives us the first really good insight and first glimpse of the gene function. We're also interested in gene expression regulation, so microRNAs are a big theme for us. And the tool set to combine expression and microRNA analysis and the functional annotation, I think is very nicely developed in this software package. And so we're really using this as a first really screening and tool set to annotate our data sets. I think we're looking for a number of diseases where we have whole exome, whole genome sequence analysis. I think we really want to very comprehensively analyze the genes which we identified. And we also want to very comprehensively analyze the impact of these genes, how that plays a role in different individuals, given the different genetic backgrounds. So we want to, really annotate that. And ultimately, I mean, the goal certainly will be to take this information, take the understanding of disease to develop new treatments, potentially, driving towards like drug development.
QIAGEN.
Sample to insight.